Hello, I'm Angela, and I'll be presenting on the microRNA in airway smooth muscle cells and how it is the most responsive to high stretch in association with cell mechanics modulation. The study was done by Wen and the rest of his team. So, some background before getting into it. Human airways continually experience varied extents of mechanical stretch, which plays essential roles in both physiological processes such as airway structural formation and functional maintaining, and pathological processes such as ventilator-induced lung injury or VILI. In fact, VILI is a common risk in using mechanical ventilation to provide life support for acute respiratory distress syndrome patients, such as the patients that are critically ill from getting COVID-19 and they have been put under mechanical ventilation, which is widely reported to be having them subjected to high stretch, which is having strain over 10% in the lungs. And these people that are under mechanical ventilation tend to end up developing VILI, which then contributes to the high mortality rate of the patients that undergo mechanical ventilation. And in some of the other studies that have been done, research into the molecular pathways involved in VILI have yet to find an effective way in reducing this mortality rate, which has led to an interest in the role of stretch in changing biomechanical behaviors of airway smooth muscle cells, or ASMCs, and the associated regulatory signaling via non-canonical molecular pathways, such as microRNAs. This is because ASMCs are known as the major mechanosensitive cells in the airways and lungs and have been reported to respond to stretch by changing contractile and secretory phenotypes in respiratory diseases such as asthma. Therefore, it is hypothesized that ASMCs may also play a crucial role in responding to high stretch during mechanical ventilation and mediate the pathogenesis of VILI. So, for the methods of this, they took two strains of primary human ASMCs that were grown in standard culture conditions with supplemented media. These cells were then seeded into collagen-coated flexible plates and serum deprived for 24 hours before the experiment. To mimic mechanical ventilation stress, some cells were stretched at 13% strain for 48 hours, while others were kept in static conditions as controls. Then the total RNA was extracted from ASMCs. The microRNAs were reverse transcribed using a stem loop cDNA synthesis kit and quantified via real-time PCR. For mRNA, cDNA was made using a first-strand synthesis kit and also analyzed by PCR. The expression levels were normalized using internal control genes, U6 for microRNA, others for mRNA, and analyzed. After the microRNA, LET7A 5P were introduced into ASMCs using lipofectamine 3000, and after 12 hours, the medium was changed and the cells were cultured for another 48 hours before further experiments. For cell viability, culturing for 48 hours, a CCK8 assay was used to measure it. The transfected ASMCs were incubated with the CCK8 reagent, and absorbance was measured at 450 nm. Cell shape changes after transfection were tracked using live cell imaging. The aspect ratio, length to width, of cells was measured for cytoskeleton analysis, and the cells were stained for imaging with confocal microscopy. Cell stiffness was evaluated using optical magnetic twisting cytometry. Magnetic beads were attached to the cells, twisted with a magnetic field, and the cell's resistance to the twisting, a measure of stiffness, was quantified. To assess how much force ASMCs applied to their surroundings, cells were plated on soft gels with embedded fluorescent beads. The bead movement before and after cell detachment was tracked using microscopy, and force was calculated using specialized software. Cell migration was also measured. It was measured in two ways. Horizontal migration, which was a wound that was scratched into a confluent cell layer, and cell movement into the gap was monitored over time. And then also vertical migration, where the cells were placed in a transwell chamber. After migration through a membrane, the cells were stained and counted under a microscope. So, getting into some of the results from this experiment, by using one of the primary human ASMC cell strain, only 16 microRNAs were found to have a cycle threshold value below 36 in them, which indicates that these 16 microRNAs are more abundant and physiologically significant in the airways. The expression abundance of each microRNA in this group was measured as fold change of the expression related to that of U6, which was the reference gene in the cells. The bar chart in figure A shows the fold change of 16 microRNAs without high stress. The pie chart in the top right insert shows the percentage distribution of the top 8 highly expressed microRNAs of over 1% in the ASMCs. And the chart in figure B shows the fold change of 16 microRNAs in ASMCs with high stretch. And it showed that LET7A 5P turned out to be the most responsive to high stretch, and that therefore the rest of the study they focused solely on LET7A 5P and its effects on biomechanical behaviors of ASMCs. To explore the effects of LET7A 5P on ASMCs, LET7A 5P mimics or inhibitors were transiently transfected into ASMCs to either up or downregulate the expression of it in the cells as compared to their counterparts transfected with scramble genes. This data confirmed that in ASMCs and LET7A 5P transfection induced upregulation in the same order of magnitude while not impairing the viability of the cells in culture. Figure A shows the ASMCs transfected with LET7A 5P mimics or inhibitors were observed for 24 hours with time-lapse live cell imaging. Interestingly, downregulation of LET7A 5P turned most ASMCs into long worm-like shapes resembling the freshly dissociated ASMCs that are considered more relevant to the morphology of ASMCs in vivo. Since cell morphology is determined by cytoskeleton, especially the stress fibers, the effect of LET7A 5P on the distribution of stress fibers in ASMCs was evaluated by immunofluorescence analysis, and it was found that the downregulation seemed to significantly increase the stress fibers' length compared to the control groups, which can be seen in figure B. And then the cell length, width, and the ratio of cellular length to cellular width, the cell aspect ratio, were analyzed as shown in figure C. This shows that upregulation of LET7A 5P led to not much change in cell length, but significantly increased cell width, resulting in significantly decreased cell aspect ratio. However, downregulation seem to show the opposite effect. And then, much like the morphology, the study found that LET7A5P upregulation significantly increased cell stiffness, whereas LET7A5P downregulation significantly decreased cell stiffness. Where figure A shows cell stiffness of ASMCs transfected with LET7A5P mimics, inhibitors, and scramble controls, and figure B shows the quantified traction forces of the cells transfected with LET7A5P mimics, inhibitors, and scramble controls. The traction force in a single cell was measured by observing the cell contraction-induced displacement of fluorescent beads embedded in the underneath elastic substrate, inhibited distinct patterns of force distribution when the cell was treated in different conditions. Specifically, the LET7A5P upregulation significantly increased traction force, whereas the LET7A5P downregulation significantly decreased the traction force. Since both cell stiffness and traction force are determinants of cell migration, they examined the effects of LET7A5P on migration of ASMCs in terms of both horizontal migration by wound healing assay and vertical migration by transwell assay. The quantitative results for horizontal migration showed that in 24 hours, LET7A5P upregulation significantly inhibited horizontal cell migration, whereas the downregulation significantly promoted it. And in the vertical migration, the quantitative results showed that the upregulation decreased it, whereas the downregulation increased the number of ASMCs that migrated to the bottom of the transwell chamber in 24 hours. Together, these results suggest that LET7A5P negatively modulates migration of ASMCs. For mRNA expression, the data showed that compared to the scramble control groups, LET7A5P upregulation enhanced mRNA expression of SMA by about 1.7-fold and MLCK by about 2.5-fold, whereas the downregulation inhibited that of SMA by about 0.7-fold and MLCK by about 0.07-fold. The figures show the fold change of mRNA expression of MLCK and SMA and ASMCs after treatment with LET7A5P mimics in Part A, inhibitors in Part B, and related scramble controls in both of those. Furthermore, in ASMC's LET7A5P upregulation clearly inhibited the mRNA expression as shown in figure C, and the LET7A5P downregulation enhanced the mRNA expression of these molecules as shown in figure D. So in the discussion, they talked about how LET7A5P was the most responsive to mechanical ventilation simulating high stretched at 13% with expression increased by nearly tenfold as compared to others by less than fourfold. This suggested that LET7A5P may be the predominating stretch response microRNA, which following that revealed that upregulation of it increased cell stiffness and traction force as well as SMA/MLCK expression, but inhibited cell migration both horizontally and vertically. In contrast, the downregulation of it resulted in the opposite effects on ASMCs. Such alteration in stiffness and force generation of ASMCs may contribute to decreased lung compliance and corresponding deterioration of VILI. Furthermore, the results revealed that up or down regulation of LET7A5P also decreased or increased cellular length and cell aspect ratio, which interestingly, the morphology of ASMCs with LET7A5P downregulation was very similar to that of freshly isolated ASMCs, which suggests that LET7A5P may have a critical role in regulating the morphogenesis of ASMCs. In addition, it was shown that up or down regulation of LET7A5P led to inhibition or promotion of mRNA expression of subfamily members of integrin and talin. This suggests that LET7A5P specifically interacts with these molecules to regulate ASMC morphology and mechanics. Overall, the results indicate that high stretch could significantly increase the expression of LET7A5P and ASMCs. This seemed to suggest that stretch-induced high expression of LET7A5P during mechanical ventilation might also play an anti-inflammatory role in lung pathology while adversely regulating stiffness and force generation of ASMCs to reduce lung compliance. These findings provide an alternate mechanism responsible for high stretch-induced effects on mechanobiology of ASMCs, but also potentially a novel therapeutic target for a high stretch-related lung pathology such as VILI. However, with this study, there were some limitations. So one limitation of the study is that it could not confirm the primary mediator of LET7A5P in regulating morphology and the mechanics of ASMCs. In addition, in the study, they examined some of the key players in the cytoskeleton dynamics and focal adhesion, including SMA and MLCK, integrin, and talin, and found that the mRNA expression of these molecules was associated with that of LET7A5P. However, the analysis did not find a direct target of LET7A5P on MLCK or SMA, which implies that there is an indirect regulation of such. In addition to integrin and talin, there are many other cytoskeleton correlated molecules that were not investigated in this study, and also that they did not investigate the specific regulatory mechanisms through which high stretch modulates the microRNA expression of LET7A5P. And other microRNAs that are abundant in the lung, especially other subfamily members of LET7A, may also participate in regulatory of morphology and mechanics of ASMCs, but these weren't studied in this experiment. In the future, these would need to be studied in order to fully understand the role and associated regulatory mechanisms of high stretch in lung homeostasis and an injury.